Good morning, everyone. We're happy to welcome you to SPG's Pulse Agronomy Workshop. My name is Allison Fletcher, and I am a Research Project Manager for Saskatchewan Pulse Growers, and I will be your host for today's session. This is the third time SPG has offered the Pulse Agronomy Workshop, and we're glad to have you join us today. For those in attendance today, if you provided either a CCA or a CCSD number at the time of your registration, you will receive credits for today's sessions. After the workshop concludes, a recording will be available online, so be sure to watch for an email from SPG on how to access those recordings following today's session. At the end of the presentations, there will be a live question and answer discussion. So if you have any questions as you listen to today's speakers, please type them into the questions box on your GoToWebinar control panel, and we will address them at the end of the speaking session. There are also handouts and resources relevant to today's sessions that you can access right through the GoToWebinar in your dashboard. SPG is guided by the grower-elected board of directors, driven to create opportunities for profitable growth for Saskatchewan pulses. Two of SPG's 5 key result areas relate to research, development, and extension, with the purpose of increasing yields of established pulse crops and promoting the adoption of new pulse crop options. This year's Pulse Agronomy Workshop focuses on 2 topics that support both of these key result areas. Today, we're going to cover root rot diseases, and tomorrow, integrated pest management and fertility in pulses. We recognize the value that agronomists bring to growers in Saskatchewan, as you are key in spreading the technical knowledge and widening the reach of this information to growers. So thank you for taking the time to be with us today. For our first session, Dr. Marie-Laure Pilet-Nayel and Dr. Lyndon Porter will walk us through root rots through an international lens, demonstrating the global root rot situation. Dr. Pilet-Nayel is the Director of Research at France's National Research Institute for Agriculture and the Institute for Genetics, Environment, and Plant Protection. She has expertise in quantitative genetics and genomics, marker-assisted selection, and disease resistance in plants. After a postdoc period at the United States Department of Agriculture's Agricultural Research Service, Dr. Pilet-Nayel managed research programs in genetics and genomics of Aphanomyces resistance in legumes for the past 20 years, in tight collaborations with pathologists, physiologists, and breeders. She has developed scientific collaborations with the United States on root rot diseases in legumes for more than 15 years. Dr. Pilet-Nayel's presentation was co-authored by Dr. Anne Moussart and research and development scientist at Terre Innovia in France. Dr. Porter is a research plant pathologist with the United States Department of Agriculture, Agricultural Research Service located in Prosser, Washington. He has been conducting pulse crop research for 17 years, primarily focused on root rot and virus issues in peas, chickpeas, and lentils. He received his doctorate degree from Washington State University in potato pathology, but converted to pulses after he was enlightened. He was born and raised in Provo, Utah, and has 5 children that he hopes are trying to make the world a better place. First up will be Dr. Pilet-Nayel, followed by Dr. Porter. Welcome.
Okay, good morning, everyone. I would like first to thank the organizers for the invitation to talk today. So my name is Marie-Laure Pilet-Nayel. I'm a researcher geneticist at INRA in France, and I'm going to present you today our research advances for pulse root rot management in France. This presentation is co-authored with my colleague Anne Moussart, who is a pathologist working at the French Technical Institute for Oilseed and Pulse Crops, and who has been working on root rots of pulses for more than 20 years. Anne apologizes today for not being able to present today, but she will be available by email if you have any questions.
Marie, sorry to interrupt. Can you just move your GoToWebinar panel out of the way? There's that green arrow. If you click it, perfect. Thank you so much. Sorry to interrupt.
Okay, sorry. Oops, no.
It's all good now. We can see everything on your screen.
Okay. Thank you. So first, I would like to present you some data about growing areas of grain legumes in France. Pea has been the most cultivated pulse crop for more than 30 years, but growing areas have dramatically declined since the mid-90s, especially due to the yield instability of the crop generated by root diseases. Faba bean, chickpea, and lentil are minor crops, but in the recent years have shown a slight increase of growing areas for their interest in providing proteins in agroecological cropping systems. These grain legume crops have suffered from root diseases, especially damaging on pea, lentil, and chickpea, on which they represent a major constraint. On pea, Aphanomyces utices is the most frequent and damaging pathogen. Fusarium are also often isolated, but we do not know exactly their level of pathogenicity and impact on yield. Pythium and Rhizoctonia are also present, but less frequent. On lentil and chickpea, Fusarium are suspected, but we do not know which Fusarium species and if other pathogens are involved. On faba bean and lupine, root rot diseases are less frequent. Fusarium are often isolated, and we also find Pythium and Rhizoctonia. So the management of root rot diseases on these grain legumes involves avoiding infested soils, managing crop rotations in sowing conditions, and choosing resistant and tolerant varieties when available, and also using seed treatments when available. So a field survey of root diseases of grain legumes in France has been initiated in 2021 for 3 years by Terre-Innovia. The objective of this survey is to identify the targets, the main pathogens involved, and their host specificity for a better management. Plant and soil samples were isolated from fields with root rot symptoms. Information was collected about crop history of these fields. A diagnostic on the root pathogens present was obtained using DNA tests, and pathogens were isolated on selective medium. In 2021, 48% of the field with root rot symptoms sampled were grown with lentil, and the most frequent root pathogens isolated on the lentil samples were Fusarium species, mostly Oxyspora, and Phytophthora has also been observed and may be an emergent problem in France. Finally, very little Aphanomyces utices was isolated on lentil. The root pathogen isolates will be tested for their virulence and aggressivity on legumes, and an analysis of the complex of root rot pathogen present in the fields will be conducted with regard to field histories. So a field survey of root diseases of grain, the field survey has been carried out since this year, because until now, efforts have mainly focused on for 20 years on Aphanomyces root rot of pea, which has been the major issue of long-term and multi-actors collaborative research programs in France. The disease has first been reported in France in 1993, following the intensification of the spring pea crop since the 80s. And it has become the most damaging root rot disease of pea and has strongly contributed to the decrease of French pea growing areas. So research has focused on this disease since 1995 with a strong collaboration between French pathologists, geneticists, and private breeders. Tight collaborations have also been developed between France and USA, and especially USDA, to develop research programs on root rot diseases of pulses. Main advances have been obtained in epidemiology for prophylactic and cultural management. First, assessing the risk before sowing is recommended by determining the inoculum potential of the field. A new online tool has been recently developed called EVA, Evaluation Aphanomyces Risk, available on the Terre-Innovia website. So in this tool, you have to provide information about regional area, rotation type, soil type of a field, and the tool will indicate if you have a low or high risk of growing pulse in the target field. A risk lower than 5% or higher than 60% to have an inoculum potential higher than 1 on a 0 to 5 scoring scale. If you have a low risk, you can cultivate, you can grow pea. If you have a high risk, you have to verify the inoculum potential of the soil before sowing, and using a biological test. Second, choosing the sowing date. Winter pea or spring pea, according to the inoculum potential of the field, is also possible to avoid the disease. We have shown that susceptibility to the disease is higher at young plant stage than at older plant stage. So when climatic conditions are favorable to the disease, March or April in France, winter pea is at a flowering stage, whereas spring pea is at an emergence stage, and the disease is more damaging on spring pea. So winter pea could partially escape Aphanomyces root rot and could be really cultivated, grown as an alternative crop in the field, in infested fields with low to medium inoculum potential. Third, choosing legume species and varieties in rotation with pea is also recommended to reduce the disease development. We tested more than 250 French varieties for their resistance versus susceptibility to Aphanomyces root rot in controlled conditions, and a very high variability was observed between species and even between varieties. Some species like lupin, chickpea, were probably non-host since we could not observe any symptoms. Others like faba bean and soybean were very resistant. Pea, lentil, and alfalfa were partially resistant to susceptible, and clover and vetch were very resistant and or partially resistant to susceptible, depending on the species. So according to the inoculum potential of the soil in the fields, growing different legume species can be recommended. If the inoculum potential is lower than 1, and if there was no susceptible legume grown in previous rotations, then there is no restriction to grow pea or any other legume. If susceptible legumes were grown previously in the field, then growing non-host or resistant species varieties is recommended, like if the inoculum potential of the field is higher than 1. So genetics of resistance to Aphanomyces urticus in different species is not well known except in pea and the main legume cultivated in France, on which we have focused our efforts to provide advances on genetics for breeding of resistant varieties. First, 2 main programs were developed to identify partially resistant germplasm. The first one was the screening program of 1,700 Pisum accessions for resistance in controlled conditions, with best accessions validated in field contaminated conditions. A few best accessions with partial resistance were identified with a root rot index score of around 2.5 on a 0 to 5 scoring scale. The second program was a recurrent phenotypic breeding program from French private breeders using resistant germplasm from the US. This program resulted in the selection of improved agronomic type breeding lines with higher levels of partial resistance, with a root rot index score between 1 and 2. We also conducted different programs in very tight collaboration with USDA to identify resistance QTL in different partially resistant germplasm using linkage or genome-wide association analysis in several populations. These programs resulted in the identification of closely linked SNP markers at consistent QTL on several linkage groups of pea genetic maps, especially a major QTL on linkage group 7, AEPS 7.6, at which we identified a common allele associated both to an increase of resistance and root area and a decrease of root losses after infection. We have fine mapped this QTL in a small interval spanning about 1 megabase on the pea genome, and work is under progress to identify genes underlying this QTL. A second major QTL, AEPS 4.5 on linkage group 4, was also identified for resistance to a specific pathotype of Aphanomyces not identified in France, but in the USA and probably in Canada. We also developed a marker-assisted selection program to introgress these QTLs in several genetic backgrounds. A total of more than 200 near isogenic lines were created, carrying resistance alleles up to 0, 1, 2, or 3 main QTLs. And the alleles were evaluated in different conditions for their effects on the disease in growth chamber, in greenhouse, in networks of field-infested nurseries, and also in networks of the field-infested versus healthy plots, yield plots. And the result showed that we could validate a QTL effect in these yields, especially QTL combination, including the major QTL. The durability of partial resistance conferred by these QTLs was also a main topic of our research. We developed a program to look at the genetic variability of the Aphanomyces urticas populations in France. Here, a total of more than 200 isolates of Aphanomyces were sampled in different regions of France, and we showed that a low genetic diversity was observed, but a diversity in aggressiveness of the isolates was observed on legumes, and especially on pea near isogenic lines. And as here you can see on the graph that on the NEAL carrying the major QTL AEPS 7.6, about half of the Aphanomyces isolates sampled showed lower aggressiveness, but the other half showed a higher aggressiveness. So we found that this QTL had a mid-wide spectrum of action against the French Aphanomyces urticas populations in sample. So the hypothesis is that this QTL could exert a selection, growing varieties carrying this QTL would provide selection of more aggressive Aphanomyces urticas populations. So to test this hypothesis, we conducted an 8-year experimental evolution assay in an infested field sown in 2016, and we grown single crop successions of pea meals and also alternate successions of pea meals and faba bean. And we will look at the evolution of the inoculant potential and the evolution of Aphanomyces urticas populations in this field experiment. So all this progress in genetics that we have provided have resulted in the recent years in the first tolerant varieties release in France. It was a really huge progress for us in France, and 3 French varieties with better yield preservation were released in and with better yield preservation in highly contaminated conditions. And a scoring scale of yield preservation in highly contaminated conditions was created, and these varieties called Caractère, Cagnotte, and Poséidon had a score of 3, meaning that their yield was more highly preserved compared to Cayenne, Astronaute, Safran susceptible controls in very highly contaminated conditions. So to conclude, I just would like to mention that really to manage Aphanomyces root rot, an integrated disease management approach is really required to better control the disease, and a mix integrate disease resistance and root architecture, rotations, cultural practices will be indispensable to apply these three combinations of practices will allow to decrease the soil inoculum potential and to reduce root rot development and damages. And we think that since in the past 20 years now, we have a less impact of the disease on pea production today compared to the past 20 years, because we think that we have a strong impact of communication on rotations and cultural practices, but the impact of the recent resistant tolerant varieties is not visible yet, maybe because of the level of partial resistance still partial and maybe insufficient, or maybe because it was these resistant varieties have been it's too early to evaluate the impact. So just to finish, I wanted to point out a very important collaboration that we have developed for 20 years between America and Europe. This collaboration network, which really allowed us to have progress on root rot of pulses. This network is now called the Root Rot Diseases of Pulses International Research Network, or RUPI network. It's a transatlantic network between France, USA since 2000, and also with Canada since 2015. And we have exchanged meetings. We have, since 2000, we have developed 11 collaboration meetings, 8 international workshops, and more than 30 co-authored publications or communication in Congress. And so we are, the network would like to address different topics, the evolution of inoculum potential, the pathogen population diversity, how the resistance loci are conserved between legumes, the genetics of multiple root diseases, and integrated management of root diseases. And so we really would like to continue these exchanges of science and and develop more work together. We will have the 8th International Legume Root Diseases Workshop on next August 2022. It will be a virtual meeting, and we also would like to promote this workshop, which is organized by this network. So finally, I would like to thank all the contributors, all the colleagues who contributed to the work, and especially PhD students and also many people in France and in the USDA who contributed to the work presented. Thank you very much for your attention.
Good morning. It's well, depending on where you are, the time of day it is, but I'm Lyndon Porter. I'm a research plant pathologist with the United States Department of Agriculture, and it's good to be here with you. Today I'd like to talk to you about managing root rot on pulses with a U.S. perspective. And can individuals see my mouse?
Yes, we can.
Okay, so first of all, I just wanted to show you the different root rot pathogens that we are dealing with in the United States, and this is a similar complex in France as well as up into Canada. Down here in the corner, this is a picture of Pythium seed rot. Pythium causes root rots and seed rots. Primarily, the Pythium seed rot is problematic across all of our pulses, and when I talk about pulses, I'm talking about chickpeas, lentils, and peas for our area. Chickpeas are extremely susceptible to Pythium seed rot. but also susceptible to Pythium root rot caused by Pythium irregulare group 4. So as far as Pythium seed rot is a problem across all pulses, and as far as Pythium root rot, it's a major issue in our peas and our chickpeas. Pictured here is Rhizoctonia root rot. You can see the infection here. It's a brown, a brownish color to the root rot that takes place on the pulse roots. This is a major problem. It's out of all the root rots, this actually is one of the least problematic pathogens that we have, but it still can cause some issues in some of our areas where we have no-till practices. It tends to be a major. It tends to be a bigger issue in our peas, and in our peas we have 3 major what are called anastomosis groups that are impacted by Rhizoctonia, and it's AG4, AG2-1, and AG8. Pictured here is Aphanomyces root rot. We only have issues on Aphanomyces root rot in our peas and in our lentils, but this is the #1 major pathogen of all of the root rot pathogens that we deal with respects to how devastating it can be on our pea and lentil crops. It can take down entire fields and cause major issues, whereas these other pathogens we're able to either manage them or they're not so severe that they'll take down and prevent the crops from even yielding. Over here is pictured Fusarium root rots. The Fusarium root rots are a major problem across all of our pulse crops, and they don't tend to take the crop down completely. We don't have a complete crop failure due to our Fusarium root rots, although it can happen on occasion. But mostly it's just it's a nagging disease that prevents symbiotic relationships with rhizobium, so we don't get as good nitrogen fixation, and it just tends to reduce yields. So we're trying to identify genetic resistance to be able to manage our Fusarium root rots. So this is another pathogen that hasn't been really classified with root rots, which is the focus of this topic. But we, in our surveys in lentils and in peas, we have found that Fusarium oxysporum, which causes what's called a wilt, is also causing a root rot in our pea and in our lentil production areas. And so this is just a look at this particular wilt pathogen. Oftentimes it'll cause a discolored vascular tissue in the root system, but we've also seen that it's causing root rot across the different pulse production regions, as I said, in our lentils and in our peas. And so we're beginning to turn some major emphasis in looking at Fusarium oxysporum as a problem in our pulse production areas. This is just a look at our a 2016 survey of our pulse production areas for peas, lentils, and chickpeas across the United States. So you can take a look at where our major pulse production areas are. They're majorly focused in Montana and North Dakota. And we've got in southeastern Washington and into northern Idaho is a major production. We're also in Nebraska and Colorado and Wyoming. We're starting to see increased pulse production as well. As a general rule, our pulse production is increasing in the United States in different areas as they're customizing different pulses and different resistant genetic varieties to different regions in the United States, and it's really taking off, particularly because of the of plant-based proteins that the pulses have in them, and there's lots of different regions that are interested in growing the pulses to have plant-based protein sources in the different states to be able to extract these plant-based proteins from for a lot of different products. So this is a look at the different cultural practices and the different management strategies that we are trying to use to manage root rots in pulses in the U.S. So I'm going to talk about different cultural practices that we're looking at and utilizing here. One of our major emphases is to look at seed treatments, and we're working closely with different chemical companies to develop different seed treatments that will manage these different root rots. As far as foliar chemicals, there has been the growers in our areas are less interested in foliar applications to manage root rots. One of the reasons is that oftentimes it requires an additional application that they're not interested in making. Although we try with some of our research that we're doing with foliar applications, we try to time them with insecticide applications that they're already making or herbicide applications that they're already making, and hoping that the tank mixes are compatible. And so that is one area we've looked at and in the past, particularly using phosphorus acid to try to manage Aphanomyces root rot. But the growers have not been interested in the additional costs of that product, as well as having to tank mix those mixes in with other things that could potentially impact the efficacy of other products they're trying to apply. We have looked at biological seed treatments, and we have not had excellent success with biological seed treatments. And the growers in our areas, we don't have or recommend currently any biological seed treatments to the growers. And none of the growers are really using biological seed treatments to manage root rots at this time. It's just been very difficult to have those biologicals work well when introduced into the soil. And here in the bottom with genetic resistance. Genetic resistance is one of the major, is a major focus that we have to try to manage root rots on our pulse crops. There are a lot of different efforts that we're making, and I'll talk about some of those later here in the presentation to try to manage these Fusarium root rots. Genetic resistance is the most economical as well as the most environmentally friendly mechanism that we have to manage our root rots. So as far as root rot management is concerned, it is critical for us that we have disease, that we conduct excellent disease surveys. One thing that I've been impressed with up in Canada are the excellent disease surveys that have been conducted in Canada across the different growing regions. And in our area, we look to do these disease surveys because this is the scouting report that we need to know how to really tackle these root rot pathogens. There are, for example, with Fusarium, with our Fusarium root rots, there are multiple species of Fusarium that cause root rots, and we need to be able to identify which species are the primary species that are causing those root rots so that we can cater our fungicide, our seed treatments specifically to manage the number one species that we're trying to deal with. And as well as our genetic resistance to that specific species. So it's critical that when we do these surveys, that we do it across different soil types, different precipitation zones, and different areas that have different crop rotations. All of these things impact the different diseases, the different root rot pathogens that develop in the soils. So in the United States, we've conducted some excellent pea root rot surveys across North Dakota, Montana, and we have some older surveys that were conducted in Washington. We, as far as our lentil root rot surveys, we just are recently completing a root rot survey where we primarily focused on Fusarium root rots that were impacting pea production areas across the United States, and we've been able to gather some excellent information on that with regards to Fusarium avanaceum being one of the probably the primary Fusarium root rot pathogen that we need to focus on in lentil, and we also know that that's a primary pathogen we need to focus on in pea. But also there's some major issues with Fusarium oxysporum in our lentil as well as in our pea that we need to address. The Fusarium oxysporum doesn't seem to be as aggressive as Fusarium avanaceum, but it's very abundant, and a high number of our plants are infected with Fusarium oxysporum. As far as chickpea root rot surveys, we have not had a lot of problems with chickpea root rot problems in the U.S., except that has changed over the last 3 years. And over the last 3 years, the growers have shortened their chickpea rotations, and they've been growing a lot more chickpeas. And because of that, we're experiencing a lot more chickpea root rot issues and major issues that have been damaging to the chickpea crop. So as far as our root rot management strategies and looking at the cultural practices that we use, I want to talk about crop rotations. I want to talk about tillage practices, no-till versus till, and rolling versus non-rolling the soil after planting. And so I'll discuss those cultural management practices. So first of all, just crop rotations. And I want to talk to it, and Marie-Laure talked about this with regards to Aphanomyces root rot and had some excellent information. But crop rotations are critical, and long and extended crop rotations where you, for example, with lentils and peas, where you at least have a 3-year rotation before you come in with a lentil or a pea again, is critical. We have had in areas where they've had short crop rotations, which are 2-year rotations with either lentil or pea, many of the growers have lost the ability in certain areas to even be able to grow to grow peas because they've grown them in too short of a crop rotation. And Aphanomyces root rot has caused a major issue to the point where they can't grow particularly peas in certain regions in our area. So as far as the crops that we grow and their resistance that we've identified, lentils are highly susceptible to Aphanomyces root rot. Fava beans tend to be resistant. Chickpeas tend to be resistant. Spring peas are susceptible, and winter peas are also susceptible, but it is what Marie-Laure talked about, is that they are at a more mature state when they are infected by Aphanomyces, and so they can survive the attack a lot better. But knowing this, we also tell our growers that there's really no difference between lentils and peas in your rotation. Aphanomyces impacts those two crops the same, as well as other root rot pathogens, and so we don't really consider lentil and pea a rotation, and that it's not a good rotation because of how similar the pathogens, the root rot pathogens, are responding to lentil and pea. So with regards to tillage practices, a lot of our growers use no-till tillage practices where they leave the stubble of the crop on the field, and they'll directly seed into that stubble. So in areas where we have no-till, no-till practices, we tend to have higher levels of Rhizoctonia root rot. And Rhizoctonia can cause these stunted patches in our pea production fields, primarily in our pea production. And so we have these stunted areas where the plants, they mature faster than the surrounding crop, and they're also stunted. So they mature earlier, but they're stunted and can cause problems with setting the header of your planter to harvest the crop, but also they just don't yield well. So in looking at these patches, they've been associated with Rhizoctonia called Anastomosis group 8. And so this can be a problem with no-till, because there tends to be higher organic matter in your no-till areas than in your conventionally tilled areas. And that higher organic matter tends to favor Rhizoctonia. So that's just something that the growers need to realize. There's great benefits to no-till, but recognizing that Rhizoctonia can be a bigger issue in their fields. So rolling practices, growers will plant their pulse crops, and then they'll roll. The purpose of the rolling is to smooth out their fields so that they have a level field to harvest their pulse crops during harvest, and it's also to improve the contact of the soil with the planted seed. The issue with rolling can be, depending on the soil, the soil content, the moisture content in the soil. When you roll, you can have, if that field is extremely wet, depending on how wet it is, determines on how compacted that soil can become when it's rolled. And so rolling can be good to level out that field, particularly for lentils that can be very short in stature, and so it can be difficult to harvest them if you don't have a level field. But Fusarium root rots, as well as Pythium, are a greater issue and a bigger problem in our soils that are compacted. And so just realizing that that as you do roll and observing the different soil moisture levels that are in the soil can determine the level of compaction that takes place, and those things can exacerbate Pythium and Fusarium root rot issues in your fields. So as far as root rot management strategies for chemical control, this is a major area of focus for us, is to test and to try to identify different seed treatments that can work to manage the root rots. And so as far as Rhizoctonia in our area, to manage that, Sidaxane has been an excellent product. The market name on that in the US is Vibrance. There's a fairly new product called Trebuchet that has showed excellent promise for managing Fusarium. It's called Pilet-Nayel, and it has helped to manage Fusarium root rot, particularly in peas, is where I've tested it for the company, and it has shown some excellent results. We are in the process of looking to test this product across the different Fusarium species to see how well it manages the different Fusarium species. In the state of Washington, our principal Fusarium is Fusarium solani. In Montana and North Dakota, one of the principal Fusariums is Fusarium avinaceum. This is on pea. And in Washington, on our winter peas that we plant, the major Fusarium pathogen is Fusarium redolens. So depending on the location and depending on the type, a winter pea versus spring pea, the major Fusarium pathogens are different that are causing the problems in our fields. So as far as metalaxyl resistant Pythium, up here in the corner is a picture of a chickpea production area in the U.S. located in the Washington and Idaho area. But these patches that you see here, where there was poor emergence and poor stand counts, we had metalaxyl resistant metalaxyl resistant Pythium develop in these fields. And so we had to do some testing to try to find a product that would work well to manage the metalaxyl resistant Pythium. And so in our studies, we identified that ethoboxam, the active ingredient ethoboxam, has been excellent. And last year it was registered for peas, which is good because we do have metalaxyl resistant problems in our pulse production areas. And there's a new product that's going to be coming out for the pulses, picarbotrazox, which is also going to be a product that's going to help manage metalaxyl resistant Pythium. And so it's good news that we do have products to manage those problems. As far as Aphanomyces, we have tried a number of different products to try to use that could be utilized as seed treatments to manage Aphanomyces root rot. The only product that was highly effective is a product with the active ingredient hymexazole. Hymexazole is used to treat beet seed for Aphanomyces root rot in beets. And it's also effective in peas, but it's too cost prohibitive. The cost of the product is so astronomical that it's not cost effective for us to be able to use it as a seed treatment to manage Aphanomyces in lentils and in peas. So as far as our major effort is focused on managing these root rots through genetic resources, looking at different germplasm, different lines of pulses across collected from across the world. This is a look at different pea genetic resources that we've utilized from across the world. You can see the different pea types that are there, and we screen all of this. We screen all of these different. There's located in Pullman, Washington, is the Western Regional Plant Introduction Station that houses world collections of peas, chickpeas, and lentils, as well as other legumes. And we utilize these resources constantly to screen for genetic resistance to these different Fusarium root rot pathogens. And then we work with our breeders and let them know which lines are providing the greatest genetic resistance to these different root rot pathogens. And then they work to integress the genes through traditional breeding into the different lines that we're trying to develop resistance for. This is just an example of a screening test that was recently conducted by my lab, just screening a whole bunch of these genetic resources in peas for Fusarium root rot. And so two of the majors, so Fusarium solani, that's over on this side. This is just this is a control for this particular line. So we always have control plants that are non-inoculated. This is a look at Fusarium solani infecting this particular line. And over here is a look at Fusarium avanaceum impacting this particular pea line. So we've screened a large amount of different lines from all over the world for resistance to these two Fusarium pathogens. As I said, in Washington, we're focused on it for breeding specifically for the state of Washington. We want to focus on lines with excellent genetic resistance to Fusarium solani because that's the Fusarium root rot that's in Washington, Idaho. However, in Montana and North Dakota, we want to focus on lines that are genetically resistant to Fusarium avanaceum. Fusarium avanaceum is much more aggressive. You can see how more aggressive it is than Fusarium solani in this particular line, but in general across all of our lines with different isolates of Fusarium avanaceum compared with Fusarium solani, we have Fusarium avinaceum as a more devastating root rot pathogen. So that's bad news for our North Dakota and Montana areas because that's the major pathogen there. But the good news is that in our screening, we have screened for genetic resistance. We've screened, this is a look at 444 lines that we screened from the Western Plant Introduction Station in Pullman that I talked about. We screened cultivars as well as these lines for resistance to Fusarium avinaceum. And the good news is that also, although Fusarium avinaceum is much more aggressive, we have better genetic resistance in our world collection to Fusarium avinaceum than we do to Fusarium solani. So even though it's more aggressive, we've got genes and genetic resistance out there that's better, that will protect the roots better than what we've been able to identify in Fusarium solani. So this is just a look at the top 10 world lines that we screened for resistance to Fusarium avinaceum, and we know their origin, and so we can go back to these areas and collect. We can have germplasm, what are called germplasm explorations to these different areas, to these different countries, and try to collect more germplasm from those areas when we realize that that there's resistance from those plants from these areas. And so that's important for us to know the origin of where they're located. It's also important for us to separate these into flower color, because your pigmented flowers tend to be feed type or Austrian winter pea type, and your yellow seeded and your green seeded food grade peas, for example, are for human consumption. And so we will look at the top. We'll rank these according to the top colored flowered as well as the white colored lines, with the white flowered lines being the lines that are going to be easier to breed for food grade purposes for human consumption. And so that's also important. Some of the resistance to these different root rot pathogens are tied to a purple flower, and we can't separate those from our white flowered. And that's sad because our white flowered are for human consumption, but we separate these into the two different groups, and we identify the most resistant lines within the purple flowered and our white flowered varieties for either feed consumption for animals or food consumption for humans. As far as root rot management strategies, there's other things to consider and that we've identified that we need to pay attention to. And one is we found that herbicide applications can also impact our root rot resistance in our different crops. Pictured over here is a chickpea cultivar. I was called out to a chickpea field to take a look at it. There was some major root rot pathogens in the root rot in this chickpea field. You can take a look at this root rot that's taking place by Fusarium that was causing a major root rot issue in their chickpea field. They were trying to figure out what they can do in the future with seed treatments or with different cultivar of chickpea that they could plant to try to avoid this devastating root rot that was taking place on their chickpeas. But as I looked around and as I surveyed the field, there was a certain section of the field where the herbicide applicator had missed. And he had not applied the herbicide that was applied to the chickpeas in that section. And the chickpeas in that area, there was very limited root rot on the roots. The plants were a nice, lush green color, a healthy green color. As you can see, these were chlorotic, and they were about 4 inches, 5 inches taller than the other plants that were in the field. And so we just realized that we need to pay attention to, even though we need our herbicides to be able to manage the different weeds that are problematic in our pulse fields, we also are going to begin taking a look at how the different herbicides impact or cause greater root rot issues in our fields, because we know that they can stress the plants out. And in this particular instance, there was such a huge stress factor on the plants that we ended up having a major root rot pathogen that otherwise wouldn't be a problem in the field.
Dr. Ford, we've just got about a minute left here, if you don't mind.
Okay, and then the last is just nematode pressure. We've also identified that nematode pressure with root lesion nematodes and pin nematodes in lentils can be major issues, that it may look like you have a major root rot pathogen issue in your field, but what it is you have a major nematode issue in your field, and the nematodes are causing damage and allowing other things to come in and cause root rot. And so these are the different things that we need to pay attention to and that we're looking into here in the US to manage root rot problems in pulse production. And with that, I'll take questions at the end of the workshop.
Thank you for your presentations, Dr. Pilet-Nayel and Dr. Porter. As a reminder, if you have any questions, please type them into the questions box and we'll address them in the panel discussion at the end of today's workshop. Next, I'd like to welcome Dr. Syama Chatterton from Agriculture and Agri-Food Canada, who's going to share an update on her Aphanomyces and Fusarium research. Dr. Chatterton joined Agriculture and Agri-Food Canada at the Lethbridge Research Centre in July 2011 as the Pulse and Special Crops pathologist. She was the first to identify that Aphanomyces root rot was present in Alberta and demonstrated that the disease was widespread and devastating on both pea and lentil crops across the Canadian prairies, and has been on a journey of discovery of this challenging pathogen ever since. She is also trying to decipher the root rot complex and the interplay between the numerous pathogens involved in that complex, and how this can translate into a robust decision support system for pulse production. She will present on current research results of field trials that are evaluating various agronomic practices that may mitigate effects of root rot in pulses. Welcome, Dr. Chatterton.
Okay, good morning. I hope everyone can see and hear me. Oh,
Look good. Thanks, Syama.
okay, thank you. So I'm happy to talk about root rot on peas. It's my favorite topic and I really enjoy being here this morning. So I'm just going to start with thanking the team that works with me. It's a bit of a different presentation for me today. I'm really trying to focus on presenting new data that we've collected in the past 2 years. And I think everyone knows it's been a challenging past 2 years, so I really want to give a shout out to my lab group. Up here at the top, I have them listed. Technicians Eric and Christine do all the field work, and Anthony Erickson does all of the lab work. And then we have 2 graduate students as well that are in our team that are working on root rot. So just a shout out to them for how hard they worked over the past 2 years under very challenging conditions. I have a number of collaborators. I'm going to be presenting a lot of field trial data. So we have sites all across the prairies and just listing the collaborators that have really helped with these trials. And then lastly, the funders. And you can see we have quite a large group of funding. There's a lot of research going on into root rot. So I'm going to start with kind of talking about the overall research vision for root rot management. And I was really happy to see that Marie-Laure ended her presentation with this because it really shows that we're on the same page across the globe on how we're looking at managing root rots. And the first is starting with a soil test that's really based on reliable pathogen quantification. And we also want to use that to be able to discover root rots early so we can look at patch management before they spread throughout the whole field. The second is, once we know we have a problem, how do we manage that disease? And Lyndon just gave a really nice overview of all the different options that we have. And some of the things that we're looking into particularly are kind of the interval period between pea and lentil crops, and also trying to develop an ooze spore decay model so we know how quickly we're losing viable ooze spores in the field. And then all of this will lead into using partially resistant cultivars. But in order to deploy those partially resistant cultivars, we need acceptable pathogen levels, or that inoculant potential that Marie-Laure was talking about in the soil. And we also need good germplasm and good lines with resistance. But I'm going to spend most of the talk today talking about managing disease, partly because the last webinar that I gave for Saskatchewan Pulse growers really focused on the soil test, and that webinar is still available as a video for people to get that information. But just a quick kind of overview on soil tests, because we have some interesting new data to present. I think everyone that's heard me talk before knows that I'm passionate about trying to get to this idea of risk prediction, and that we want to be able to test the soil for the presence and quantity of soil-borne pathogens and then compare those to thresholds. And the last talk that I did give at SPG, I really focused a lot on what those challenges are with trying to develop a precise and accurate DNA quantification system for soil. So just giving a little reminder here from that last presentation, how we're looking for these little tiny spores in this huge amount of soil. They're clumped, it's kind of a challenge to know if you're actually going to be collecting soil that has ooze spores in them. So because of that, I kind of say here it's an imprecise science. We're dealing with these tiny microscopic organisms in these huge amounts of soil. As well, the nature of ooze spores, you can see them down here. You can see that really thick cell wall makes them resistant to sort of the common DNA extraction technique. So that's where a lot of that difficulty lies. And then as well, each soil type does have a different threshold dose. So I think when Marie-Laure showed kind of the, what did she call it? The evaluation for the risk of Aphanomyces, that flow chart there, I was happy to see there that it talks about the region and the area that you're in, because I think that really plays into what soil type you have and what your environmental conditions are. And so ultimately, my goal, I'll consider my career done when I have released a decision support system to provide to farmers based on pathogen quantification, soil properties, and field history. But in order to get there, we need more reliable and rapid high throughput method for Aphanomyces, and that starts including some of the other pathogens. And we're just starting to work on a collaboration with a couple of scientists from AFC at Saskatoon that really have expertise in sort of these rapid high throughput methods. So I'm really excited about that new project that's just about to start. And then also we need to look at integrated tools for multiple pathogens. I think Lyndon really did a great job of outlining all the different pathogens that are involved in these root rot complexes. And then when we're dealing with multiple pathogens, we see that they have different soil and environmental conditions that are their optimum. So that's what I'm going to talk about next, some exciting new research results from a grad student. We really struggled with this, shut down over COVID most of 2020, and then trying to ramp up research in 2021. And so we finally have some good results for this. And what we're looking at is how soil moisture contributes to disease when we have both Aphanomyces and Fusarium avinaceum. And this is really a lot of work. You can see here these pots, and the grad student will come in every day, measure the soil moisture levels in these pots with these probes, and then water accordingly. And what we were trying to target was low. So that's below about 30% moisture, so drought conditions. Medium is about field capacity, looking at about 40% moisture, and then high would be saturated conditions at all times, so usually between 50 to 60% soil moisture. And we're looking at how Aphanomyces, Fusarium avinaceum develop, and then also in combination with each other. And I think what's really remarkable is how well these results turned out. I know people hear us talk all the time about how important soil moisture is, and here we can see that we really didn't get any Aphanomyces root rot developing until we were under those high saturated soil moisture conditions. Fusarium avinaceum was interesting because we see disease under low, medium, and high, and not really any difference. But then I think what's the most remarkable here is when we look at that combination of Aphanomyces and Fusarium avinaceum, even at the low drought condition, we see we get very high disease levels, much higher than either pathogen alone. And then under the high saturated conditions, we can see disease was the highest. And then we also looked at sort of colonization levels of these pathogens on the roots. And again, here we can see with Aphanomyces levels here, really didn't get any substantial colonization until we were under those very, very saturated conditions. Interestingly, Fusarium avineseum was the opposite. We really saw high colonization levels under those low or drought stress conditions, and then it actually decreased as we went up to higher soil moisture. I didn't show what the colonization looked like in this combined treatment because it didn't really make sense. So I have to kind of look at those results closer and figure out what's going on, because actually both pathogens had lower colonization levels. So there's some interplay going on there. So really what we find is that soil moisture is essential for Aphanomyces root rot development. It doesn't seem to be as important when Fusarium root rot development and colonization. However, root rot was the worst when both Aphanomyces and Fusarium were present across all soil moisture types. And I think that's why we see that root rot can develop even under kind of the drought conditions that we saw in 2021. What's very interesting is that the Aphanomyces biomass was high only in the roots from saturated soil. And so that's when we're going to see these massive increases in inoculum to the soil. So I think the last presentation that I gave at SPG webinar, we went into a lot about soil, or about cropping history and the environment the last time you grew peas. And so this is really nice lab data to support that we're seeing that you have that problem under really high saturated conditions and the problem persists. The other thing we looked at was temperature and disease, and I should mention that for this experiment, this was done under highly saturated conditions. We unfortunately don't have the resources to look at temperature and soil moisture because of the fact that you have to be in there watering those plants every day. Is just too much of too much labor input when we're still under COVID restrictions. So we just looked at under highly saturated conditions, and I think you can see right away that there really was no effect of temperature from 18 up to 28, a slight decrease at 28 degrees, but still really high disease levels. And then when we look at colonization by Aphanomyces and Fusarium, we see slightly higher levels of Aphanomyces at the cooler temperatures. And then we did see a drop off in the lateral roots of Aphanomyces colonization at 28 degrees, but we're still getting good colonization of the taproot. And for Fusarium, again, at about 23 degrees, 18 to 23 degrees seem to be optimal for colonization. So this results weren't quite as exciting as the soil moisture. We did see that temperature under saturated water conditions is not as important as soil moisture. There's a slight trend towards higher colonization by Aphanomyces under these cooler conditions. What I would really like to look at is temperature by soil moisture by soil type. I should mention all of this trials were done in from the dark brown soil zone, so a fairly high clay content soil. And I suspect we would see kind of different results if we were looking at a clay loam or silty loam soil, and then to be able to do temperature by soil moisture. But like I said, those are really, really labor intensive experiments. So that's all I wanted to talk about soil testing and what we're moving towards, more precise data on how soil environment is playing a role in risk management. And now I'm going to move over to managing disease. And Lyndon again went over very well what we know on how to manage disease, and really our number one factor here is avoidance or extended rotations is our only management options. Like he said, we have seed treatments that claim to provide early season suppression, but we don't see them providing any prevention or long-term reduction of root rot over the growing season. Alternate pulses like faba bean, chickpea, and soybean are good rotational crops. I'm going to talk about some of our research with that a little bit later. And then, like I mentioned, our crop history and environment are really our most important drivers. So you get that recipe of water plus oospores equals disease. And so when you've grown crops in a really, really wet season, your risk sort of increases and it carries over for a number of years. So some research that we're currently doing looking at managing disease in the field is listed here. So we're looking at cover and rotational crops. pea intervals and the inclusion of alternate pulse crops in that rotation. Also looking at lime soil amendments, intercropping with brassicas and maybe some other species of interest like oats, and then also evaluation of seed treatments and natural products. And Lyndon talked about this last one a lot, and really that it's been challenging to find anything that works against Aphanomyces root rot in the field. So I'm going to talk primarily about our results from these three research trials here. Okay, so for cover rotational crops, this has been a challenging trials that we've had to sort of modify as we went through, partly because of dry conditions. We were originally looking at shoulder season cover crops, either pulled off. Peas can come up pretty early, and then we were thinking, okay, well, what if we can get some brassica cover crops on there in the last 6 to 8 weeks of the growing season? We just couldn't get establishment. So we moved it over to looking at kind of a full season of either cover or rotational crops. So what we did, this started in 2019, we included rotational crops that would be taken either to seed or terminated early and used as a green manure, or we evaluated some crops just as cover crops only. And we did this at 3 locations in Lethbridge, Lacombe, and Saskatoon, also with and without tillage. And then in 2020, the peas were planted into these the 2019 footprints of these crops. So really it's kind of looking like at a rotational study or following what crops are maybe good to follow, or peas good to follow after. Then we looked at root rot severity, pathogen levels, and yield. We wanted to measure oospore levels in the soil, but it's, like I said, it's been challenging because of what I talked about earlier. This is what the cover crops look like in 2019. Lots of different brassicas. We have oats, we have rye, long plots that we till half and then don't till the other half. So we're looking at both till and no till. And then once these are taken down and harvested in 2019, then we follow them up with a pea crop in 2020. So what did we find? First, there was no effective tillage, so that was just removed completely from any of the analysis. And we saw no effect on disease severity. Basically, it was high and uniform at all treatments at all sites. Here's an example of what it looked like at Lacombe. 7 is the max for our disease severity, and we can see that most of these are close to 7. We saw the same thing at Lethbridge and Saskatoon. And I should say 2020 was quite a wet year, which was unfortunate, maybe not the best year to be evaluating pea root rot, unlike 2021. We all have our challenges. But when we look at the pea yield, I was actually very pleasantly surprised by the results that we saw in Lacombe. And so here we have our pea, this would be pea on pea stubble, pea grown on canola stubble, and pea grown on cereal. And then these are a couple of the crops that we're evaluating: a caliente mustard, fava bean, Synapsis alba, white mustard, and then oats. And here we can see a nice significant increase in yield when peas were grown on caliente mustard as a green manure stubble, fava bean either on green manure or as seed stubble, and then oats on green manure. And here we can see almost like a doubling of the yield here. So I was very excited by these results. And then when we look at just the green manure crops here, we had a couple of forage, blend 1, blend 2 listed down here. We can see that we had a significant increase in yield in Lacombe on stubble grown on this blend 2, which is really a whole bunch of different forage brassicas basically, and then not really much of a difference in any of the other crops. So Lacombe yields were very exciting. That's why I showed them first. Then when we look at Lethbridge, we didn't quite see the same results, nor in Saskatoon, so I'm not going to show the Saskatoon results. But what we did find was that on mustard, rye, clover, fava bean, and caliente mustard seed, here the pea yields were higher than what were grown on the cereal stubble or pea on pea, but really not any significant difference when grown on canola stubble. So this is just, just a rotation effect. Doesn't seem to matter what crop is maybe there, although we didn't see the same thing with oats, even though we saw a really good response in Lethbridge. And then when we looked at the green manure crops, again, we saw some effect of this peas grown on this blend here, which is a whole bunch of different brassicas, significantly higher than the cereal or pea control, but not than the canola control. So the next steps are repeating this, seeing if we can see those really nice results that we saw in Lacombe. And so we grew these cover rotational crops again in 2021, and then we'll grow peas in 2022. The big problem is that 2021 had a very disappointing establishment of biomass, so we might not see that there's enough biomass there to have any effect. This is a picture of our fava bean crop at our Lethbridge site. It was really destroyed. There's pea leaf weevil damage, drought effects, and then I think right there you can see grasshoppers, lots of grasshopper damage. We even sprayed for grasshoppers twice and it really didn't make a difference. And unfortunately, most of our plots looked like this. So if we don't see an effect in 2022, I think it's because we really didn't get a good establishment of our rotational or cover crop. So next I'm going to talk about the pulse break and inclusion of alternate pulses. And what we're looking at here is a frequency of pea with and without an alternate pulse crop and rotated with cereal and canola. And the alternate pulse crops that we were looking at were chickpea, fava bean, and soybean, each at different locations. And we sort of chose the crops that were best matched to that area. So I'm going to just go through the results for Tabor and Redvers. I think it would be a really long presentation if I tried to go through all the locations. And what I want to start with here is our Tabor. We last had lentils at this site in 2016. So we had a 1 year break where we grew peas in 2018. And here we can see the disease severity is about 4.6, yields at about 1,300. Then we went and grew peas again on these plots. So we had a 1 year break between the peas, and we can see big increase in disease severity, really horrid yields, and I'm going to get to that when I talk about the 3 year break. A 2 year break, we did surprisingly well, actually. So lentils were last in 2016, and then we had peas here in 2019 with a chickpea in between. I think it was grown in 2018 as well. And so we see actually a surprising reduction in disease severity and a nice bump in yield. So that was interesting after the 2 year break. Then we get to the 3 year break where we would expect to start things looking better. But if you remember, 2020 was an extremely wet year, particularly in Tabor. And so we saw that disease severity bump right back up, and we really saw these really bad yields. I will say this is partly due to weeds. Because of the COVID restrictions in 2020, we couldn't have students come out and weed the plots, and we had really, really bad thistles in these plots. And so this is kind of a weed effect as well as the really, really wet effect. And then when we had the 4 year break here in 2021, quite dry year, we can see that the disease severity has come down from what it was, but we are not seeing that why we got such a low disease severity after that 2 year break in 2019. I really don't have a reason for that. We don't have the yield results yet. And then comparison to what we saw in Redvers, and this is where we rotated with soybean crop. They last had peas in 2015. We started the trial in 2018, so we were already at kind of a 2 year break. And then here we can see, again, fairly high disease severity, the 1 year break, high disease severity again. And then we saw similar to what we saw in Lethbridge, where we had here as a 3 year break because the last peas was in 2015. We see this really nice reduction in disease severity. Yield didn't really improve here, but it actually improved a lot when we had soybean included in the rotation. And then we look at the 4 year break. Redvers wasn't as wasn't as wet in 2020. So we see that reduction in disease severity sort of carry over. And then we haven't seen yields really improve. And then with the 5 year break in 2021, we're still seeing that reduction in disease severity, but we don't have the yield data yet. And we saw similar patterns at the other sites. In Lacombe actually has stayed high the whole time because it's been wetter site. Swift Current, we saw kind of a similar reduction, and Saskatoon is fairly similar to Tabor. It had very similar weather conditions. So, here it's kind of showing, well, it's hard to say how long you need that pulse interval, and it really seems to be location specific.
Dr. Chatterton, just wanted to let you know we've got about 4 minutes left here.
Okay, so I'm going to end off here just talking about some of the liming trials for Aphanomyces. The first thing we did was look at it under greenhouse conditions, and then the plan was to start some trials in 2021, but again, because of COVID restrictions, it got delayed, and we just are going to start it in fall 2021 rather than the spring. And so what we're looking at is a spring versus fall application. product type and rate. We are tilling the product into the soil, and then we're going to kind of follow soil pH and calcium, and then disease severity, yield, and oospore levels in the soil. So I've shown this data before, just reminding everyone that we saw some really nice results from hydrated lime in the greenhouse. No, we didn't see the same with quick lime or zero grind, just different formulations of lime. But we saw this really nice reduction in disease severity as we increased the lime rates. So we took, oh, here's some pictures of what this looks like. We can see really, really nice, or a lot less disease symptoms compared to our control here. So what we did in 2021, because we couldn't go out and do the full field trial just because of issues with getting product and co-op students in the lab, we did what we call the microplot trial. So we went out and buried these pots in the soil, these large pots, and then they get filled with infested soil mixed with lime products. And the lime products we used were hydrated lime and zero grind. And we had some pretty good, interesting results. We measured both Aphanomyces and Fusarium. Oftentimes you can't separate them, but in this trial, they were really, really nice separation of the symptoms. And so we can see that Aphanomyces was reduced with all of the hydrated lime rates from low to high, and this was significantly lower than the control. Interestingly, Fusarium was increased compared to zero grind, actually reduced Fusarium levels, and so we see a significant difference between these. So I guess the challenge of preliminary data is showing that there's a benefit of hydrated lime, but maybe a trade-off with Fusarium root rot. Zero grind lime is not as active as hydrated lime, so it might take longer to release the calcium, and that's why we're looking at a fall application as well to see if it takes more time to sort of equilibrate. And we did start these trials out in the field in fall 2021, and lime was applied a couple weeks ago at many of the sites. So finally, for managing disease, I think what we need to know, what we're looking at, just started a trial as well, burying a whole bunch of roots out in the field, in the soil, to see what happens with the oospores. We're trying to look at oospore decay periods. As well, we've come across some suppressive soils where we don't seem to get Aphanomyces to be able to establish. So we're trying to figure out what's going on with those soils, and then also looking at what are some of the host triggers of pathogen germination. And really, we're trying to just discover any agronomic practice besides field avoidance that will actually mitigate root rot, and that's what we're chasing here. Just very briefly, the last minute here, talk about partially resistant cultivars. I think both Lyndon and Marie-Laure talked about this a lot. This is being worked at primarily at the CDC at the University of Saskatchewan. I do say that resistance to Fusarium species may be more challenging, but that's because we're dealing with a lot of different Fusarium species in our trials. And here's just a germplasm line that we evaluated out in our disease nursery, and we can actually see a lot more. It's got some resistance to Aphanomyces, but a lot really bad Fusarium root rot. So, you know, maybe not all lines are going to work as well. And so we're looking at other ways of protecting resistant sources, and how do we find new resistant sources, because this is going to be a continual battle against root rots. And so perspectives for managing root rot, I think we all know it's challenging. There's lots of research going on. I hope my presentation helped to show that. I really believe that the foundation is knowing what's in your soil and coming up with that soil test that we can accurately predict your risk. Combine that with disease management strategies and improved resistance. So really a multi-layered approach to managing disease. And so with that, I will take questions at the end of the session. And I always like to leave with a picture of my hikes, because it's easier to summit mountains than it is to do research on root rot. And with that, I will end my presentation.
Thank you again, Dr. Chatterton, for your informative update. It's always great to hear from you, sort of periodically, to hear where your research is at, and your enthusiasm really shows. So we're grateful to have you working on this problem for us. Just a reminder, any questions that you have for Dr. Chatterton or for any of the speakers this morning should be sent to us via the question box, and we'll address them in the Q&A portion of our session at the end. Next, I would like to introduce Dr. Sabine Banniza, who is here to share her research into root rot in chickpeas and the chickpea health problem in Saskatchewan. Dr. Sabine Banniza is a professor of plant pathology at the Crop Development Center of the University of Saskatchewan, Canada, where she has been working on pulse crop diseases for more than 20 years. Much of her research has focused on understanding host pathogen interactions of pulse crops and their implications on population dynamics, resistance breeding, and disease management in pulse crops. A considerable amount of research has concentrated on foliar fungal diseases. For the last 9 years, her group has also been working on emergent root rot pathogens, particularly Aphanomyces and Fusarium, with the primary focus to identify durable resistance in pulse crops. Welcome, Dr. Banniza.
Thank you, Alison, for the introduction. And I guess we'll switch crops a little bit because I will be talking about chickpea. Now, both or all Marie-Laure, Lyndon Porter, and Syama have given you a great background on root rot problems, and you will sort of see similar themes coming up with the chickpeas as well. So, if you have been dealing with chickpeas in the past as a grower or as an agronomist, you will have noticed that we have a problem there as well. And it was quite interesting to hear Lyndon talking about chickpea root rot problems popping up, because this is what has been happening here as well. Now, we noticed increasing root rot problems in our breeder plots probably around 2017, 2018. So actually had applied for this project before it really blew up in commercial fields here. So it was probably around 2019 that sort of problems really showed up in commercial chickpea fields, and there were reports from fields where that there was literally a meltdown of the chickpea crop. And although we don't know yet definitely what the issue is, sort of the problem has been tackled from several angles. So herbicide damage, as Lyndon Porter already pointed out here as well, there is sort of a sense that herbicides may play a role in there. There was a question about whether we are also seeing resistance breakdown to ascoghyta blight, but then clearly there are also more root rots. Probably, or it is assumed that some of this is due to microbial pathogens, but there are also reports of insect larvae and also nematodes. So it's definitely sort of a multi-cause issues, which doesn't make it simpler. So I just want to sort of pop through some of the different pathogens that have been associated with root rot, specifically in chickpeas, in addition to maybe Rhizoctonia and Pythium, which are sort of already very familiar. So as you have heard throughout all the presentations this morning, sort of Fusarium root rot is a very common theme, and Fusarium root rot are obviously present in chickpeas as well. Lyndon already mentioned that Fusarium oxysporum, Forma specialis ciceris, is a big issue in some of the US, but worldwide it is the main Fusarium species on chickpea. Fusarium avineum, Solana, Redolens, and Sporotrachioides have also been reported from chickpea. Whether and what pathogen is really affecting our chickpeas is still a little bit in the unknown. Another pathogen that has been reported from chickpeas in the past is Thelaviopsis or Rhizoctonia basicola. There was a report on chickpea in Washington in 1985, so we decided, okay, we'll have a look at this pathogen as well. Yet another one is charcoal or dry root rot caused by Macrophomina phaseolina. I couldn't find a picture on chickpea, so I'm presenting you here with a picture from bean. It was reported on chickpea in California in 1974. It's fairly common in India, and it's promoted by hot and dry conditions, which would apply maybe to a certain degree to our chickpea growing regions in the south. Another pathogen or group of pathogens is verticillium, which causes a wilt, verticillium wilt. We are primarily talking about two species here, Verticillium alboatrum and Verticillium dali. Verticillium alboatrum has been reported again from California in the 1950s. And in fact, Verticillium dali was reported from canola in Alberta in 2 years ago. So it's definitely a pathogen or group of pathogens that is very likely in our soils. Another group is Phytophthora root rot, and Lyndon Porter already mentioned this pathogen as well. It's primarily Phytophthora medicagenis that we are talking about here. It's a well-known pathogen of alfalfa in North America, but based on my conversations with Pilet-Nayel, our forage breeder here, it doesn't seem to be very common in Saskatchewan. It's a very common disease in Australia, and the pictures I'm showing here are from Australia, and it has a big impact there on chickpea production. So that's why we decided, okay, maybe we should sort of include that as well. So Lyndon sort of emphasized how important it is to do disease surveys, and this is always sort of the beginning of a journey when you're dealing with a new disease problem for disease management, but also and very importantly for disease breeding, because you need to know what pathogen you want to breed resistance against. So surveys were conducted specifically for chickpea root rot in 2020 and 2021, which were organized through the Saskatchewan Ministry of Agriculture in collaboration with the Saskatchewan Crop Insurance Corporation and the Saskatchewan Association of Rural Municipalities. And I can't thank these organizations enough to sort of support these activities, because without them, it would be very, very hard for us to do that, in particular with the changed rules about accessing farms and agricultural lands. So in 2020, when we started with this, we decided to sort of spread our net quite widely. So we asked these growers to identify one field in each of the 42 rural municipalities where chickpeas are grown. So the agrologists went out there, did a visual assessment of the plants, and then sampled the roots for us. Because of COVID, sort of there was a little bit of a delay between sampling and us receiving them, so we decided to freeze them, and then we got the frozen samples. Now that did not allow us to do a disease rating on the roots, but we were then able to use them to do molecular testing of the roots to identify the samples. So from each of these fields we got 5 samples with a visual rating on the above ground biomass. This year it was a little bit easier because of the relaxation in restrictions based on COVID, and we had also changed our strategy a little bit. So once we sort of had an idea about how widespread the problem is, we decided to sort of focus on the most affected areas. So we picked 19 of the worst affected rural municipalities, and in these 1 to 3 fields were evaluated. And sort of similar to 2020, a visual assessment of the plants was done. Roots were digged up, they were shipped to us, and then we could do a visual assessment of the roots here. And then similar to 2020, we then processed these roots for molecular testing and also did some isolation. So first the 2020 survey, and I apologize for this big table here, but I wanted to show all the data. So the severity of above ground symptoms, so that's the shoots, ranged from anywhere from healthy plants to almost dead plants. So maybe you can see it, there are sort of some rural municipalities where there were areas with dead plants, but overall sort of moderate to maybe moderate levels of disease on the shoots. Well, when we then proceeded to doing our molecular detection, we did not detect any Fusarium culmorum, Fusarium sporotrichoides, and Fusarium oxysporum, verticillium, or Phytophthora. What we did find was Fusarium solani and Fusarium redolens in particular, and I'm showing this here in this maybe somewhat complex table where in the rows you have the different rural municipalities, and then each column represents a Fusarium species, Fusarium solani, Fusarium redolens, Avenacum, and sporotrichoides. I've color coded the cells. So if you remember, from each of these fields, 5 samples were collected. And so the darker the color, the more of these samples tested positive for this species. So in 2020, probably sort of the most prevalent pathogen was Fusarium solani, followed by redolence, and then Aphanomyces and Sporotrichoides. There was one field where we detected the Laviopsis, and interestingly, it was our research farm. But I guess it's good news that it's nowhere else. In 2021, as I indicated earlier, we reduced the number of rural municipalities that were sampled, so there are just 19. And these were specifically selected because based on the 2020 survey, we know that there were more root rot problems in those rural municipalities. And you can see that disease severity was a little bit higher, sort of the minimum maximum range was the same, 1 to 5, but the mean value on above ground shoots was a little bit higher than in 2020. Ranging from 2.2 to 4.4. You also notice that we did a mean root disease severity score here and had some samples where we only had small lesions, so very little disease, all the way to 100% root discoloration and hypocotyl lesions here in rural municipality 7, which had the worst disease rating. When we then look at the spectrum of pathogens that we found through molecular detection, Fusarium solani, Aphanomyces, and redolence, and the first 3 columns here were again the most prevalent species. Maybe sort of the severity had shifted a little bit compared to 2020. 2021 obviously was much drier and much hotter, and that may have affected which species was more present in these roots. We also detected Fusarium culmorum and Fusarium sporotrichoides in some fields. So this was different from 2020, where we didn't detect this. We also, and this was a little bit surprising, detected Verticillium dali in several fields, and more commonly than I would have expected based on the 2020 results. So that's probably something where we should sort of have another closer look, because this could potentially be a quite serious pathogen. But we did not detect any Fusarium oxysporum, which is good news because it is the main pathogen in other chickpea growing areas. No Phytophthora, which is also good news because it's sort of in the same group of organisms as Aphanomyces. So the management of this pathogen is much more complex than a pathogen like Fusarium. No Verticillium alboatrum, and this year we also didn't detect any Laviopsis again in commercial fields. So just as with the other pulse crops, it seems like Fusarium solanum, Aphanomyces are the very common and maybe very important pathogens on chickpea as well. Redolence has been recovered from other pulse crops as well, but is considered to be minor compared to solanum and Aphanomyces. So the next step for us was to initiate pathogenicity testing, and these are really the first preliminary tests here I'm showing. We used the Desi chickpea 24520 from the breeding program here. We inoculated by seed soaking, and we focused initially just on the most important Fusarium species that we had identified through our surveys. And we measured emergence, height, and disease severity here, focusing on the hypocotyl at 12 days post-inoculation. So this is a graph here showing root rot disease severity. Each species is color-coded here with bars in different colors, and you see that we randomly picked a number of isolates to get a sense of how variable the virulence is in these isolates. You also notice the Fusarium inflexum there. This was not recovered from any of the disease surveys, but we had recovered this Fusarium species previously on a chickpea root, and so we thought, okay, we'll just throw it in there. So what was very interesting to see is sort of even with just 5 isolate, the fairly wide range of virulence in Fusarium avineum. So if you look at far wide, 5, it almost had caused 100% of disease severity on the hypocotyl, well over 90%, compared to the others that were anywhere from 20 to just under 40 or just over 45% disease severity. So that was quite interesting, and probably so it makes me think, well, maybe we should look a little bit more closely at these populations, because it is very well possible that although these species are common in quite a range of different crop species grown in rotations, that there is a little bit of specialization going on during the cropping season, depending on what crop is grown. So based on these preliminary results, it looks like Fusarium avineum is maybe the prime candidate or #1 candidate for resistance breeding, but Solanae and redolence may also be important pathogens to look at eventually for resistance breeding.
Dr. Banniza, we've got about 3.5 minutes left.
Perfect, because I'm already at my conclusions.
Wonderful.
So, clearly, root rots in chickpea are widespread, and if you're growing chickpeas or are operating in those areas, you will just nod your head here. Just as in the other pulse crops, Fusarium, Solanae, and avineum are very important pathogens there, and potentially also redolence. So we will have a closer look at these 3, and then probably initiate screening with avineum if this is confirmed as the prime pathogen. Verticillium dali was also detected in 2021, so I think we should have another look at that, because that could potentially be also a big issue. So what are we doing now? Well, we will be doing more pathogenicity testing with all of the different species we have identified, and possibly also increasing the number of isolates per species. And then we'll start screening chickpea germplasm to identify sources of resistance, and sort of move that into the chickpea breeding program for root rot screening. So just to acknowledge the Agricultural Development Fund of the Saskatchewan Ministry of Agriculture for funding, and Western Grains Research Foundation, then again sort of sampling by the Ministry of Agriculture, crop insurance, and SAM, and input and cultures from Dr. Chatterton, with whom we collaborate quite closely on all of these root rot projects. And I also want to acknowledge cultures from the Canadian Culture Collection of Fungal Cultures and the CBS NOR collection in the Netherlands. Thank you very much for your attention. And just as Syama, I also want to acknowledge my research group. They have had some tough years, in particular last year with the drought and the pandemic. Sometimes it has not been fun. Thanks a lot.
Thank you, Dr. Banniza, for sharing your experience with chickpea diseases. If you have any questions for Dr. Banniza or any of our speakers from the morning, please type them into the question box now, as we're going to move into the live Q&A with our presenter panel. So I'm going to ask the morning's presenters to please turn on your cameras again. And I'm going to hand things off to Sarah Anderson, who's the agronomy manager with SPG. Sarah, you're welcome to start asking the questions.
Hello, everyone. And yeah, I just want to reiterate our thank yous and acknowledgements for such a great session. You guys jammed in a lot of a very complicated topic into a short period of time. So we have quite a few questions that I might not be able to get through all of them here, but maybe starting with Dr. Pilet-Nayel's presentation, we had some curiosities about the overall farmed acreage in France. So you mentioned that there was approximately 240 to 250,000 hectares of pulse crops, but relatively, where does that put the pulse crops in the range of other competitive crops?
Yes, thank you for this question. Can you hear me?
Yes.
Yes, okay. So really, the pulse crops are really minor crops finally, because wheat or wheat crop is more than 1 million hectare cultivated in France. Rapeseed is more than 1 million also. We would like, there is a political protein plan that has been initiated this year to try to double to double the areas, the growing areas. So to try to have really more pea, but also more other crops, pulse crops like faba bean or like lentils, like chickpea. So I don't know if my answer is if I answer well to the question, but definitely 200,000 hectares is not enough compared to the 700,000 hectares that we had in the 90s, and it's definitely compared to the other crops like wheat, like rapeseed, really low growing areas.
Okay, well, thank you for that context. I guess just maybe sort of following up on some of France's practices, you mentioned, you know, 3 varieties that had partial resistance, and these are currently available to growers.
Yes.
And what has been the grower feedback to those varieties?
I think it is too early to have the answer to this question. These 3 varieties were released to the French registration office very recently in 2019. So they have just been started to be grown, but we don't have information. We know that they are qualified as tolerant because in our French registration office procedure, these varieties have been tested on a network of field, highly contaminated fields, and they have really showed decreased the yield losses in highly contaminated fields. So, but we do not have more. We need to know. We need to have more years of growing to know, to have the feedbacks of the growers. So.
Okay. There's a bit of a follow-up to that, but I think there's a qualified panel here to answer the follow-up. Is what IPM strategies are being used or should be being used to ensure that resistance doesn't break down, either for those varieties or ones that, you know, we hopefully have coming down the pipeline in North America. Maybe start with you, Dr. Pilet.
Me?
Either or, yes. Dr. Banniza, if you have some comments.
Do you want me to answer? So I think it is important to realize, I mean, the French research that Marie-Laure presented already showed that if you grow these partially resistant varieties in heavily infested fields, you are exerting a lot of selection pressure on them. And so I think it will be important that growers understand it's not the silver bullet where you take these varieties and plant them into your heavily infested field, because that will erode the resistance very, very quickly. So a lot of the management strategies we are promoting now will still apply, but it may mean that instead of 6 or 8 years, maybe you can get back into your field after 4 years, right? But we still need a lot of gather a lot of data on this ourselves, because it's all a little bit the unknown. And maybe Marie-Laure, you can add to that.
Yes, I think definitely these varieties are not. We should grow them in not highly infested fields, but just they are recommended to be grown in low infested fields. But it's because, of course, the level of partial resistance is too low, still too low to have really to be able to grow them in highly infested fields. So just for the evaluation of these varieties, it was important that we could use a network of highly infested fields, and moreover, this network has been developed by French breeders, and they have developed each of them have developed has developed his own infested plots. So, but of course, it's too early and too complicated to recommend these varieties to be grown in highly infested fields. I don't know if I answer well to the question, but
I just wanted to make a comment as well, is that the resistant genes in the French lines are a culmination of years and years of research and screening that's happened in the US as well as in France to identify the genes associated with this. And so these are the best of the best, and there's not a whole lot of genes out there that work well against Aphanomyces root rot. And so taking care of this genetic resistance is extremely important because we don't have much to fall back on.
That's a very, very important point. There is nothing else in the pipeline right now. So
especially we recommend not to use the major QTL alone. To our French breeders, we recommend to accumulate several QTLs in a same variety, not just one. So it's quite, we have first results that show that near isogenic lines cultivated, grown in single crop. Okay, single crop, so it's not a realistic rotation, but grown in a single crop rotation are able to develop more and more root disease, Aphanomyces root disease on the roots. So because we increase the inoculum quantity in the soil, and moreover, the isolates, we have first results showing that the isolates are more and more aggressive because partially resistant genotypes can select more aggressive isolates. So we can growing partially resistant varieties could select more aggressive isolates. So we really have to use highly resistant genotypes. That it's possible but using genomic selection procedures by accumulating resistance alleles and not only one major QTL.
I just wanted to add one other thing to this, is that if we lose the genetic resistance that the French have already developed and put in, kind of our next step as far as genetic resistance is probably genetically modified organisms being introduced, and that's not a friendly approach right now, but that kind of would be the next level, just because we have not found a lot of great resistant genes to Aphanomyces root rot to be able to manage it. So
perfect, thank you for your comments. Switching gears to maybe a little bit more of in-season practices, Dr. Porter, when you had mentioned some of the impacts of herbicide residue specific to those chickpeas, there was quite a few questions just asking, what was the herbicide or the herbicide group? And was there anything specific about how it was sprayed that maybe allowed for a little bit more aggressiveness in the root rot versus the check strip?
Yeah, I still need to track down the different compounds that were sprayed on the field and visit with the technician, the field rep that managed that. I wasn't able to get the full product chemistry or mix that they had, but yeah, I mean, it's as far as just the stress. We're really limited in different chemistries that we can put on the pulses to manage for herbicide management, and so it's just it's critical that we understand what they can do. As far as there's lots of different environmental conditions that needed to be looked at for what the environmental conditions were when the application was made. And so that's important, as well as the growth stage that it was applied at. And so there's lots of different things that need to be taken a look at with regards to when we apply those herbicides, the growth stage of the plant, and the environmental conditions that are present in the field. So, and that hasn't been figured out completely as far as the impacts at the different growth stages and environmental conditions.
Perfect. You had also mentioned some foliar applications and trying to combine that with passes of herbicide application. Is there any comments that you have or practices that are showing some promise?
We have, so with regards to trying to figure out ways to manage Aphanomyces root rot, we've played around with phosphorus acid, and phosphorus, and this is phosphorus acid, not phosphoric acid. Phosphorus acid is, but we've really found mixed results. We have seen where it has worked, and we have seen where it has not worked. And some of the interesting information that Dr. Chatterton presented was just showing that the importance of early moisture, high moisture levels at the beginning at emergence, and how that can increase Aphanomyces development on the crop. And in the year where we saw excellent, what we did was we incorporated the phosphorus acid with an insecticide application that they wanted to control pea leaf weevil at that time, and so we tank mixed it with an insecticide to manage pea leaf weevil. And it happened to be a very wet season right around emergence, where we had rain that came down, and it created the perfect environment for Aphanomyces to infect the pea crop that we were working with. And we got outstanding results as far as phosphorus acid providing some protection to the root system, and significantly improved the yield of that pea crop. But we've had other places where we just haven't had the Aphanomyces pressure, and so the application hasn't mattered. It wasn't needed. The crop didn't need it, and it didn't do a whole lot. You didn't lose anything by applying it. Like statistically, you didn't lose money by applying the phosphorus acid, even if it was low pressure, but because we still got some yield response out of it. But as a general rule, the growers have not embraced it they haven't embraced it in the dryland area, but they have embraced it in the irrigated areas. And so they'll use foliar applications of phosphorus acid to manage Aphanomyces root rot in our irrigated pea fields, but not in the dryland, and because of mixed results. But that's the main foliar applications. Growers have also played around with the strobilurin, your quadris, your azoxystrobin, and some of your other strobilurins applied as foliar applications to try to manage Fusarium and Rhizoctonia, mainly Rhizoctonia root rot. But they are mainly applying those simply because they see that there's some sort of yield bump, and they haven't identified why the yield bump associated with these foliar applications. But some growers are applying them just to try to get a certain yield bump, but there's no real reason as far as research on why it's providing that yield bump.
Okay, thank you. Yeah, a few more questions we'll try and sneak in here. I guess another one pertaining to rotation and moisture. So just as a catalyst for Aphanomyces, I think it was brought up in everyone's presentation, and also avoidance in the rotation as well. So in areas that were dry this season, growers need to be concerned about tightening up pulse rotations for the 2022 growing season. And specifically, many growers are looking at consecutive lentils and peas because of herbicide options, herbicide carryover, and limited rotation options. So what do you suggest as a management strategy? Lyndon, we can start with you, but Dr. Chatterton, this was also brought up in your presentation too.
So they're looking to come back in with the same crop because of pure economics, and that's just what that's their option. So what to do as far as managing? Good luck. As far as managing Aphanomyces root rot, is that the particular question with regards to those? So really, you hope for great environmental conditions that aren't going to be favorable for Aphanomyces root rot in the different areas. So first of all, if you're looking at doing this, you can look at weather predictions. If it's going to be a very wet, then your risk is going to be extremely high. And as far as other things, there's some different. There's ethaboxum has been looked at. I think Dr. Chatterton has looked at it. I've looked at it. It doesn't provide as a seed treatment. It doesn't provide outstanding protection. It may provide a little bit a benefit, but we've been hard pressed to find outstanding management of Aphanomyces with ethaboxum, which is your Intego seed treatment. I'm not sure what it's called in Canada, what they might be marketing it as, but it's better than nothing, but it's not super good. I've looked at it in under greenhouse conditions, and it provides some protection, but it's not going to prevent Aphanomyces from. It may suppress it just a little bit, but if you're looking at options, that's better than nothing to at least have it on there. And as I said, the foliar applications of phosphorus acid have been hit and miss. And I've looked at the research up in Canada that's been done, and it's been more miss than hit with efficacy of phosphorus acid. So I don't know that I'd recommend that. Really, what it comes down to is that there can be varying levels of resistance, varying level of partial resistance, or levels of susceptibility of the different lines that you can also choose to grow. And I would just take a look. There's probably some cultivar screening that either Sabine or Dr. Chatterton have done with lentil or pea varieties. Is that true? And to the level that you can choose, I would take a look at their research to see which are more or less susceptible, because they're all susceptible, but then they'll vary in their levels of susceptibility. And I would take a look at their research results and see if you have the choice to choose, which often they may not, but if you do, to take a look at the level of susceptibility of the different cultivars and choose the ones with the best resistance, so partial resistance.
Perfect, thank you. Dr. Chatterton, do you have anything to add? And it's a bit of a loaded question. I think a lot is just wondering, it was so dry this year, did we really amplify that tightened rotation?
Yeah, it's a good question. And I think all you can do is hope 2022 is not a wet year if you're going to choose to do that. But who wants to hope for a dry year, right? So it's, I guess you kind of pick your poison if you want a drought year or a wet year. And I think, there might not have been a lot of inoculum left behind in the soil in 2021 with pulse crops because it was so dry, but we still did see root rot develop in our field trial areas that were highly infested, like at our site in Tabor, you know, which was really, really dry. There was still root rot on those peas. So it's still there, even though it's dry, it's not like it's gone away. So I think that's just, you know, the big caution is that people think they're safe, but those oospores, they'll sit in those dry conditions and they'll come back if there's moisture. So it is risky to look at back-to-back pea and lentils.
Okay, perfect. Maybe switching gears, I guess, to the chickpea front. So Sabine, you covered off, you know, just the root rots in chickpeas and as it may or may not pertain to the chickpea health issue. But are the symptoms similar, like of what we're seeing with, yeah, root rots and their symptomology? Is it also conducive to what's being identified as the chickpea health issue as well?
Yes and no. I mean, I think the root rots definitely contribute. I'm not yet convinced that it is just a root rot problem. I think there are other factors playing into it. And I mean, we know that in some fields, nematodes were found. And so as Lyndon Porter explained, maybe they are making things worse. There are suspicions that maybe there is a herbicide by pathogen interaction, right? So I'm sure the root rots are an important part of it, but I'm not convinced it is the whole story.
Perfect, thank you. Unfortunately, there is a pile more questions. You guys have certainly generated a lot of interest. I think we are getting close to the end of our time line, so I'm going to hand it back over to Allison, but I do think that we will have an opportunity to circle up and get some feedback to the attendees who asked these questions. So I will turn it back over to Allison, but thank you for entertaining the ones we were able to get to.
Thank you. Thanks again to all the presenters. Thanks, Sarah, for managing the Q&A. As Sarah mentioned, there was lots of great questions, so we will follow up with those and circulate those answers around to the attendees. The recordings from today will be posted on the website, and you'll receive an email on how to access them. You'll also receive an email with a link to a survey for you to share your feedback with us. Your opinion does help SPG deliver valuable extension opportunities and helps us to continuously improve what we do. Lastly, keep an eye out for information coming soon about SPG's winter extension meetings. We'll once again be making stops in the communities of Assiniboia, Belfort, Elroys, Etonia, and Davidson throughout January, February, and March of 2022. The meetings will be open to in-person and virtual attendees. So look for more information and registration links that will be shared in the coming weeks. And finally, we'll be back again tomorrow to discuss integrated pest management and fertility. So thank you, everyone, and have a great day. Bye.